Hi guys. Today we're going to be learning all about factors that affect the performance of enzymes. So things that can um, make enzymes work better or worse. And we're going to start with just a kind of recap on what we've learnt so far about enzymes. So your task for the next few slides is to pause at the beginning of every slide and have a look at the diagram or the picture and think about what I'm trying to portray with that picture. So often in an exam, you're going to get given a diagram of some sort, and you have to be able to interpret what in that diagram you need to use to support your answer. So have a pause here, have a look at that picture on the left and the one on the right, and think, what is this trying to say? Okay, and then come back. Okay, so looking at this picture on the left, we can see that this red strand here is a piece of DNA. So it's just simplified to look like a tube so that there's not too much going on. Each of these red things represents a gene. So one strand of DNA would have hundreds of genes on it. So again, this is just simplified. And what you can see here is RNA polymerase, that's this yellow thing here. Ase is a hint that we're looking at an enzyme. So protease, amylase, lipase. The suffix ase um, represents enzymes. So RNA polymerase, this yellow enzyme here, is transcribing. So this is part of protein synthesis, the first part of that central dogma that I discussed on the last screencast. This enzyme is transcribing a gene on the DNA into a piece of RNA. So in order for the DNA to be used, we know DNA has instructions for creating proteins, but the cell can't just read the DNA as it is. All of the bases, so the ladder rungs, are enclosed inside the DNA. So polymerase basically reads the DNA and creates a single helix that represents the gene on the DNA that is in question. And then that piece of RNA can travel to the outside of the nucleus and be picked up by a ribosome that can translate this code into making a protein. So there's an enzyme involved in reading DNA and making RNA, and then the ribosome uses the RNA and kind of pops all of the amino acids that are coded for on the RNA into a peptide, and that peptide can go on to become a globular protein. So a type of protein is enzymes. So enzymes are the product of protein synthesis. So your DNA codes for how to make an enzyme, and enzymes are involved in making the enzymes. So I mean, we've touched on how enzymes are so important and they're found everywhere. Not only do we make them in the body from the instructions on our DNA, but we need enzymes to read the DNA, and we need enzymes to stick things together at the other end. So yeah, they are the little soldiers in the body. They do everything. They make themselves. There's about 3,000 types of enzyme. So all of those 3,000 enzymes would be coded for on different parts of the DNA. So different genes would code for how to create different enzymes. And yeah, even enzymes are included in the process. So that's what we see here. I just added this in while I was paused just to help me discuss that point. On the right here is a picture. I'm not sure if it's a electron microscope photograph or if it's some sort of like CGI version, but it's a picture of amylase, which is a 3D globular protein. Amylase breaks down starch, large carbohydrate, into glucose, so small simple sugars. We have amylase in our saliva, and we also have amylase in our stomachs. So yeah, it starts breaking down your food in your mouth, as well as your teeth mechanically breaking down your food. Enzymes chemically break down your food at like a atomic level. They break the bonds between things. So you start digesting literally as you eat in your mouth. Your digestion starts there. And yeah, amylase would be coded for by a piece of RNA that is transcribed from DNA. So your body makes amylase, out of the instructions it would find for making amylase on part of your DNA, as well as, genes that would code for every other type of enzyme, not just amylase. So, yep. Enzymes are coded for by DNA. Enzymes are made from amino acids. Okay, that's what these singular purple dots are. And then as they get strung along, that creates a peptide. And then once you start curling multiple peptides together into one big ball, you've created a polypeptide, which acts as a protein. A much more complex, curly 3D version of what the ribosomes are making. So, yep, enzymes are three-dimensional proteins. So, yeah, this should all be familiar by now. Okay, we're pausing on this one here, and you're going to think about, right, what is this diagram showing me? I'll come back in a second. So this diagram is showing you that enzymes, the use of enzymes, they are responsible for reducing the amount of energy that's needed to start a reaction. So lactose you will find in dairy products, and the body needs to break down lactose into glucose, a simple sugar, and galactose to be able to use it or store it. But the process of breaking lactose into glucose requires energy. It's a really effortful process. And so, yeah, it takes a lot of energy to do that. And what an enzyme can do is basically reduce that hill. If we use the hill as a metaphor for how hard it is to do this reaction, an enzyme basically makes the hill easier to get over so that reaction can happen a lot easier. Okay, in other words, enzymes lower activation energy. So we need to start using that keyword, activation energy, the energy used to activate a chemical reaction. All right, pausing here. Let me know what you think this is about. Okay, so hopefully you've noticed by now that at no point is the enzyme used up in the reaction, whether it's putting things together or breaking them apart, doesn't actually affect the enzyme in any way. So enzymes can go on and do the same thing again and again and again. Enzymes are responsible for turning substrates, which are the reactants of a chemical reaction, into the products. So substrates to products. All right, you've got two more to look at here. So pause, come back when you think you know what it's about. All right, so the top left represents the fact that there are two significant types of enzymes. There are enzymes that take small monomers and stick them together into a larger product or a polymer. And this process requires energy to be put in. So there's an effort in making things out of smaller things. These are called anabolic enzymes. So an anabolic reaction is putting things together, and a catabolic reaction is breaking things apart. So that would require catabolic enzymes. Catabolic reactions produce energy. So when you break them apart, energy is released. Okay, an example of that is ATP breaking into ADP and PI and releasing energy during that process. Okay, and an example of an anabolic reaction, so putting things together, would be anything that is a synthesis reaction. So synthesis means making things. For example, photosynthesis is the production of food in plants. So that is the synthesis of food out of smaller reactants, and that requires energy put in. And also protein synthesis, so making proteins, ribosomes making proteins out of genetic material instructions. So putting things together into one big product. That means that you need to put energy in to allow that to happen. All right, this one here. Enzymes are specific to particular substrates. So that's really all I was getting at there. I'm going to expand on that in this slide. So pause here, think about, these are two separate but connected diagrams showing two different theories about how enzymes and substrates can come together. All right, so up until recently, scientists believed that enzymes were very specific and perfectly created to fit an exact substrate, like a lock and a key. So we called that the lock and key theory. Um, but what we've recognized more recently is that actually enzymes seem to morph around the substrate when they are binding together. So they aren't the perfect shape for each other, but the enzyme kind of morphs and grows onto the substrate when they bind together. And this is what we refer to as the induced fit theory. So to induce something is to force it to happen. So the enzyme kind of forces itself around the substrate until they finally fit together. And what's significant about this theory is that it actually supported the idea that enzymes can actually focus on a number of different substrates that are related. So you might have some enzymes that work on a couple of simple sugars that are similar but not the same. So yeah, enzymes aren't perfectly specific to just one substrate all of the time. And the last thing that you might recognize on the slide is this term here. So enzyme substrate complex is what we call the enzyme and the substrate when they are bound to one another. So while there's a reaction occurring, they are referred to as the enzyme substrate complex. Okay, so moving on to factors that can affect enzymes. So what things can make enzymes work better or work worse? Now, obviously, with that being the topic, that's kind of a hint as to the answer of this question. But yeah, just thinking about what do you think it is exactly that's happening in the body when you hear of stories of people dying from hypothermia? What is so dangerous about getting cold? What actually is the problem? All right, and here is the answer. So um, we know from our last kind of mini unit that at normal temperatures the plasma membranes that surround our cells are fluid, so we refer to it as the fluid mosaic model. They're kind of wobbly and slippery, and the membrane is kind of squishy, and the things can float around each other. So all the different things you find in the membrane, that's the mosaic part. A really important substance in the membrane is cholesterol, so lipids or a type of fat. And these are actually lipids themselves, or the whole membrane itself is made of phospholipids. So all of these are fatty acids. So yeah, that's going to be really important when we think about what happens to animal fat at low temperatures. Just before I move on, obviously we know that the function of the plasma membrane is really important because it affects how things get in and out of the cell like this. So if the membrane is affected by anything, such as temperature, that's going to affect how the cells can get rid of wastes and how they can get their nutrients. And that's going to affect every cell in the body really drastically. So when you think of a piece of meat, or the fat on something, or butter, for example, is animal fat as well, at room temperature or lower, you'll see that these animal fats are solids. They are saturated fats, which means they turn solid at low temperatures. When you heat up fat, so in a frying pan or butter in the microwave, these saturated fats become liquid, so they're able to move past each other. But if you think about it, if our cell membranes are made of fats and we get lower than body temperature, all of those fats in the membranes are going to become solid and things aren't going to be able to move in and out. Oh, that's not going to help me. In and out of the plasma membranes that surround the cells. So every cell is going to be affected by this phenomenon that all the lipids in that membrane are going to solidify, and that's going to stop things going in and out, and reactions are going to stop because there's going to be no oxygen going into your cells for respiration to create energy, and then that energy can't be used for other things. So there's a whole chain reaction of things that go really drastically wrong when essential substances can't get into cells and then the wastes can't get out. I'm going to come back in a second screencast because this is about to cut out, so I'll see you in a sec.